Greetings everyone and welcome to another exciting genetic engineering and biotechnology news webinar. Our presentation today is entitled CAR T-cell target selection using innovative in situ hybridization technology. I'm Jeff Bugaliskas, technical editor for GEN, and I'll be the moderator for today's webinar presentation. An increasing part of the cancer therapy conversation over the past several years has come from adoptive cell transfer techniques, and in particular chimeric antigen receptor, or CAR T-cells. CAR T-cells can recognize and eliminate neoplastic cells expressing specific protein antigens and has seen success where other treatments have failed. As with any new success, however, comes the realization for the limitations of the treatment. For instance, the current generation of CAR T-cells cannot distinguish between neoplastic and normal cells that may also be expressing the same antigen, often resulting in adverse toxicity events. Developing methodologies for identifying novel CAR T-cell technologies specific to cancer cells is paramount for continued therapeutic success of this unique immunotherapeutic approach. Let's meet our speakers for this GEN webinar who will review the RNAscope in situ hybridization method and show how the technology is applicable to the CAR T-cell and immuno-oncology fields. Courtney Anderson is a group leader of applications at Advanced Cell Diagnostics. Dr. Anderson will provide a brief introduction to the RNAscope in situ hybridization technology and show how it can be utilized for a variety of applications with specific attention to the visualization of gene therapy biodistribution. James Rotman is a Senior Director of Translational Development at Bluebird Bio. Dr. Rotman will discuss ways of incorporating in situ hybridization methods, such as RNAscope, into preclinical safety assessments for chimeric antigen receptors. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We'll try to get to as many questions as we can, so simply hit the Ask a Question button below the presentation screen, type in your question, and hit OK. All right, without any further ado, let's get our webinar underway. Courtney, take it away.
Thank you very much, Jeff, for that introduction, and thank you all for joining us. Today I will discuss the RNAscope in situ hybridization technology and show how you can visualize the tissue biodistribution of gene and cell therapies using this technology. The RNAscope in situ hybridization technology is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context. It is a hybridization-based signal amplification system with a unique probe design that allows for simultaneous signal amplification and background suppression. The technology consists of three parts: a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots, which can be quantified manually or with digital image analysis platforms. The assay allows for detection of multiple RNA markers that can be visualized with either fluorescent or chromogenic labels. And the assay can be performed on a wide variety of sample types, including FFPE tissues, fresh frozen or fixed frozen tissues, PBMCs, and cultured cells. The two key features of the RNAscope technology are its probe design and signal amplification. The oligonucleotide target-specific probes are depicted as Zs to emphasize the fact that they have two regions connected by a linker. The bottom of the Z complements and hybridizes to the target transcript, while the top of the Z is the base for the amplification structure. When two Zs hybridize in tandem to the target sequence, it creates a binding site upon which a preamplifier can bind and the amplification tree can be built. A standard RNAscope probe will consist of 20 Z pairs pooled together that are designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a minimum of 3 Z pairs is needed to bind to the target RNA sequence in order to generate enough signal for molecular detection. The second key feature of the RNAscope technology is the signal amplification. After hybridization with the target RNA, each Z pair creates a binding site at the upper portion of the Z for the signal preamplifier to bind. Each preamplifier can bind multiple amplifiers, and each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex at each double Z binding site. Labeled probes contain either a chromogenic enzyme or fluorophore that generates a signal detectable under a standard bright field microscope or fluorescent microscope. This signal amplification strategy allows for the visualization of target RNAs as an individual dot, where each dot represents an individual RNA molecule that can be quantified. Lastly, with the RNAscope assay, background is eliminated because the signal is dependent on two Zs binding next to each other on the target sequence. If both Zs do not bind next to each other, then the preamplifier cannot form a stable hybridization and the amplification tree does not get built. Consequently, no amplification of nonspecific hybridization occurs, generating little to no background signal. This combination of probe design and amplification system ensures a high signal-to-noise ratio. Shown here is a brief overview of the RNAscope product line. We offer the manual assay with both chromogenic detection as well as multiplex fluorescent detection with up to 4 fluorophores. And the fluorescence assay can be performed on both FFPE and fresh frozen samples. We also offer fully automated assays that are compatible with both the Leica Biosystems BondRx and Ventana Discovery instrumentation. In addition, if you would rather have us run these assays for you, we can perform the experiments at ACD using the same products and the same analysis tools I've just described through our Pharma Assay Services group. It can take as little as 4 weeks from when we receive your samples to delivering results to you. In addition to the RNAscope assays, we also offer a highly specialized assay called the BaseScope assay. This is based on the RNAscope technology and therefore is capable of single molecule detection, but there are a few differences. One is that due to a different amplification system, the BaseScope probes can detect signal with probes containing as few as one double Z pair. Another difference is that while the RNAscope assay is a robust assay to detect gene expression for most RNA targets, the BaseScope assay is specifically designed for the detection of exon junctions, short target and highly homologous sequences ranging from 50 to 300 nucleotides, and also single nucleotide point mutations. The RNAscope technology is ideal for many applications, including the detection of any gene expression provided the target sequence is known, non-coding RNAs for which antibody detection is not possible, molecules that are challenging to develop antibodies against, such as GPCRs, and identifying the cellular source of secreted proteins. The RNAscope assay can also be used to validate high-throughput data, identify the co-localization of multiple genes, and also combined with IHC to have a detection of both RNA and protein, all while visualizing gene expression with morphological and spatial resolution. The assay has been applied in a wide range of research areas, including neuroscience, oncology, and infectious diseases, in addition to drug discovery and development in the biopharma arena. In particular, today, I'd like to highlight the applications of RNAscope in gene and cell therapy and also immuno-oncology. The RNAscope ISH technology is an ideal solution to detect therapeutic or target gene expression and vector sequences in individual cells within the tissue morphological context, addressing critical questions on the efficacy and safety behind nucleic acid and cell-based therapeutics. It can be used to visualize in vivo delivery of therapeutic gene expression and vector sequences, both viral and non-viral, and identify the specific tissue, cell type, or subcellular localization expression pattern of the target. In addition, one can confirm RNAi and mRNA therapeutic target gene sequences and associated biomarker expression in specific cells or tissues. CRISPR-Cas9 and other genome editing in tissue can be verified by detecting Cas9, guide RNA, or associated biomarkers. And also trafficking of CAR T-cell therapy to the correct target site can be confirmed with RNAscope, as Dr. Rotman will show in a little bit. With the RNAscope assay, it is also possible to differentiate therapeutic gene sequence from endogenous transcripts and at single nucleotide distinction sensitivity. Lastly, the assay can be used to detect, monitor, and quantify therapeutic molecules and targets during preclinical ADME and safety assessment. And I'd like to show a couple of examples for each of these categories. The first is to look at CAR T therapy. A variant of epidermal growth factor receptor, EGFRV3, is the most frequent mutation found in glioblastoma patients. Given its prominent role in glioblastoma disease progression, numerous therapies are being developed to target EGFRV3. Researchers at UPenn, MGH, and Novartis recently published their work with RNAscope for the in situ characterization of CAR T EGFRV3 in glioblastomas. The authors used an RNAscope probe designed against the 3' UTR of their CAR design to examine the cellular localization of the CAR before and after infusion of the therapy in glioblastoma patients. One of the key benefits of using RNAscope for CAR detection is that, unlike the CAR protein, which in humans is indistinguishable from endogenous, the viral UTRs provide unique tags that the RNAscope assay can detect to specifically visualize where the CAR is trafficked to in the tissue context. And in addition to the UTRs, RNAscope or BaseScope probes can be designed to target any portion of the CAR design. In this study, in situ evaluation of the tumor microenvironment after CAR-T EGFR-V3 infusion revealed specific detection of CAR-positive cells and more interferon gamma-positive or activated T cells by RNAscope, and increased but patchy T cell infiltration by IHC. Overall, RNAscope was able to visually confirm that CAR-T EGFR-V3 cells were trafficked to the tumor site of glioblastoma patients and resulted in on-target activity. EGFR-V3 is a gain-of-function mutation that arises from an in-frame genomic deletion of exons 2 to 7, leading to constitutively active oncogenic signaling. Detection of EGFR-V3 in FFPE clinical specimens by IHC and real-time RT-PCR approaches have been developed but are suboptimal due to the lack of specificity and spatial resolution, respectively. Therefore, a robust assay to identify EGFR-V3 status within the tumor morphological context is highly desirable. Therefore, we use the BaseScope assay to identify EGFR-V3 status within the tumor morphological context. 1 double-Z pair probes were designed to span the exon junctions in order to detect either wild-type, mutant, or both transcripts. The wild-type EGFR probes span the junctions of either exons 1 and 2 or exons 7 and 8, while the EGFR-V3 specific probes span the junction of exons 1 and 8. A common probe that spans the junction of exons 8 and 9 was also used to detect total EGFR, or both the wild-type and EGFR-V3 transcripts. In this slide, we show 2 glioblastoma cases with the probes I just described. With both wild-type probes, we see staining in both cases, indicating both glioblastoma samples express wild-type EGFR. However, with the mutant probes, we only see signal in the case on the left, indicating that this case is positive for the EGFR-V3 variant, while the case on the right is not. Taken together, we can specifically identify the EGFR-V3 status in FFPE glioblastoma samples. Another area of gene therapy research is AAV-based therapy. AAV, or adeno-associated virus, has proven to be an effective delivery vehicle with high transduction efficiency, stable transgene expression, broad tropism, and the ability to achieve systemic delivery, making it a safe and efficient gene delivery method for non-dividing cells. However, it is essential to understand the biodistribution of a gene therapy product in target and non-target tissues, as well as the persistence of transgene expression in those tissues. We utilize the RNAscope assay to detect AAV transduced cells in the monkey retina. A proprietary AAV expression vector driving GFP expression under the control of the CB promoter was injected subretinally into the eyes of cynomolgus monkeys. RNAscope probes were designed to target either the CB promoter or a DNA sequence of the AAV vector or the GFP transgene mRNA. In this slide is an overview of the retina probed simultaneously for the AAV vector, shown here in green, and GFP transgene, shown here in red, with a distinct AAV GFP transduced region inside the subretinal bleb and a non-transduced region outside the subretinal bleb. In the transduced region, the AAV vector was detected primarily in the nucleus of cells in almost all retinal layers, while robust GFP transgene expression was detected in both the nucleus and cytoplasm in almost all retinal layers. Neither the AAV vector nor the GFP transgene was detected in the non-transduced region. Furthermore, specific retinal cell populations were identified using probes for rhodopsin, shown here in green, and opsin-1, shown here in red, which detected rod cells and cone cells, respectively, in both the transduced and non-transduced retinal regions, and only in the rod and cone layer. A side-by-side comparison of the AAV transgene stained image with the cell type specific stained image indicates that the rod and cone cells were transduced by the AAV vector and expressed the GFP transgene. The RNAscope assay has also been applied for the efficacy of microRNA therapeutics. Approximately 20% of patients with the neurodegenerative disease ALS carry a mutation in the gene SOD1. Borel and colleagues used an artificial microRNA systemically delivered by AAV to silence SOD1, referred to as mere SOD1, in SOD1 G93A mice, which is a mouse model for ALS. RNAscope ISH demonstrated that SOD1 expression was substantially attenuated in motor neurons transduced with MIR SOD1. You can see in the left column, in the control spinal cords, there's robust expression of SOD1 in yellow, and this co-localizes with CHAT in red, which marks the cholinergic neurons, including motor neurons, and GFP in green, which marks the transduced cells. However, in spinal cords from the MIR SOD1 treated mice, we see significant knockdown of SOD1 expression in the GFP transduced CHAT-positive motor neurons. mRNA therapy is another promising nucleic acids-based gene therapy, as it provides transient production of therapeutic proteins without the need for nuclear delivery or risk of insertional mutagenesis. DeRosa and colleagues used lipid nanoparticles to deliver human erythropoietin, or HEPO, mRNA by IV injection to mouse liver. RNAscope ISH confirmed delivery of human EPO mRNA to hepatocytes over a time course following injection. Following mRNA delivery, there was also an increased human EPO mRNA, serum protein, and hematocrit. And serum EPO protein is shown here by ELISA, following a similar trend as the mRNA in vivo expression profile. Taken together, this data shows that exogenous mRNA-derived EPO protein maintains normal activity in vivo. Finally, another key application of the RNAscope ISH assay is the combination of ISH with IHC to simultaneously detect RNA and protein in the tissue context. Despite the fact that RNA-ISH and IHC target different molecules, they can each provide valuable information that complement one another. For example, one can identify the cellular origin of secreted proteins, characterize complex tissue structures by identifying cell types and the gene expression inside those cells, and dissect the regulation of gene expression by differentiating between gene regulation at the transcriptional level and at the translational or post-translational level. Here we combine the RNAscope ISH assay with IHC to visualize immune infiltration in the tumor microenvironment. In this image, we can observe FOXP3 positive cells in green infiltrating a CD8 positive and PD-L1 positive tumor microenvironment in a lung cancer sample. I'd like to end by showing the number of peer-reviewed publications, which exemplifies the growth and adoption of the RNAscope technology. We had our first publication in 2011, and since then there have been over 1,000 peer-reviewed papers published using the RNAscope technology in numerous journals, including many top-tier ones. And the RNAscope technology is highly relevant across multiple fields of research, with over 40% of the publications in cancer research, followed by neuroscience and infectious diseases. If you would like more information on the RNAscope technology, please visit us on the web at acdbio.com. Thank you for your time.
Thanks, Courtney. That was a wonderful way to begin our webinar and a great segue into our next presentation. Before we move on to Jim's presentation, I want to remind the audience to submit questions for our Q&A session right after Jim's presentation. We'll try to get to as many questions as we can, so simply hit the Ask a Question button below the presentation screen, type in your question, and hit OK. All right, Jim, as they say, the floor is now yours.
Thanks, Jeff, and welcome, everyone. As Jeff said, my name is Jim Rotman. I'm a veterinary pathologist at Bluebird Bio in Cambridge, Massachusetts. And the title of my talk today is the use of in situ hybridization during chimeric antigen receptor target selection and preclinical safety assessment. During the course of the talk, I'll tell you how we've incorporated advanced cell diagnostics in situ hybridization method into the chimeric antigen receptor preclinical safety assessment process. I'll show you how we use in situ hybridization for target selection to help us qualify monoclonal antibodies for immunohistochemistry. I'll show you a method we've developed to track chimeric antigen receptor T cells in tissues. And we also use in situ hybridization to predict CAR T preclinical toxicology organs. And I'll give you an example of that as well. Toward the end of the talk, I'll describe preparation and quality control of tissues, cell pellets, and cytospin slides for RNAscope in situ hybridization, including some example protocols and examples of tissue quality issues. And finally, I'll tell you how we've optimized our in situ hybridization parameters and selection of control probes for each tissue, and finish with a summary. Now, for those of you who may not be familiar with the topic, chimeric antigen receptor T cells are a form of adoptive cell therapy. And adoptive cell therapy uses ex vivo cultured tumor antigen-specific T cells to treat cancer patients. Now, there are 3 different forms of adoptive cell transfer, including tumor-infiltrating lymphocyte therapy, which collects tumor antigen-specific T cells from patient tumors, grows them in culture, and then provides them back to the patient therapeutically. Antigen-specific T cells can also be collected from the patient's peripheral blood, restimulated in culture, grown, and provided back to the patient again. But the version of treatment I'd like to talk about today is the chimeric antigen receptor T cell. In this case, we collect lymphocytes from a patient, we transduce them with a synthetic receptor, grow them in culture, and then provide them back to the patient. Chimeric antigen receptors were initially described by Eschar back in '96 and have recently been popularized by Steven Rosenberg and Carl June. The chimeric antigen receptor is a synthetic receptor consisting of an antigen recognition domain, essentially a single chain monoclonal antibody, and then an intracellular T cell signaling domain, which provides both the first and second signals needed for T cell activation. As I mentioned, we collect lymphocytes from a patient, transduce the receptors into those cells, expand them in vitro, and provide them back to the patient. And CARs are currently being used to treat both hematologic and solid neoplasms. One of the biggest challenges we face today is that the current generation of CAR T cells can't distinguish between target expression on normal versus neoplastic tissues. So our goal is to select target antigens that are uniquely expressed by the patient's tumor and not expressed by normal tissue, ideally. Now, traditionally, the way to study target expression in tissue samples is immunohistochemistry. But the problem is often monoclonal antibodies aren't available for the targets we're discovering. Many of the commercial available antibodies aren't specific for their targets. There can be lot-to-lot variability problems. And so we have to painstakingly validate each immunohistochemistry reagent. This takes a lot of time and is not practical when you're trying to screen through a large number of targets. In addition, target expression may be very low, or we may not be able to pick it up with immunohistochemistry. So about a year ago, we switched over to in situ hybridization for our initial safety screen. And then targets that make it past this initial screen are evaluated by immunohistochemistry. This slide outlines the Bluebird Bio CAR T cell target selection algorithm. Our bioinformatics group screens through the entire human transcriptome for targets with high tumor and low normal tissue expression. They look at public databases, the medical literature, and so on. And this yields a reasonable pool of targets for us to look at. We take each of these targets one by one and study them via in situ hybridization on a panel of normal and a panel of tumor tissues. The targets that have an acceptable profile are then advanced in the pipeline. We then attempt to find a monoclonal antibody against the target. We start with commercial sources, or sometimes we have to make our own monoclonal antibodies. And then we use cell pellets and a panel of normal tissues to qualify this antibody by comparing the protein expression pattern to the mRNA expression pattern. Once we've identified and qualified a monoclonal antibody, then we stain up the same panel of tumor tissues that we had done previously via in situ hybridization. And ideally, the protein and messenger RNA expression patterns will match. Finally, when we have a target with an acceptable protein expression profile, we make a CAR T cell against that target and proceed with in vivo efficacy studies. These are typically xenograft studies in NSG mice. Now, the other thing we try to do when we develop the chimeric antigen receptors is to identify one that will cross to the mouse, because in that case, our study becomes a combined efficacy and toxicology study. Now, the normal tissue mRNA expression pattern that we're studying in public databases is derived from tissue lysates. So if you have a rare cell population in that lysate that's expressing your target, it's possible that the target mRNA will get diluted out and you won't see it. So this is why it's important to perform in situ hybridization as an extra level of scrutiny during target selection. And this slide demonstrates a couple targets we've rejected because of unexpected target mRNA expression patterns. Consider target B here, where we see expression in the hair follicle at approximately the level of where the stem cells will be expressed. Here's target C in the inner and outer nuclear layers of the retina. Target D popped up in respiratory epithelium, and target E popped up in enteric neurons. In addition to target expression, we also use in situ hybridization to help us qualify monoclonal antibodies for use as immunohistochemistry reagents. And the big picture is that we stain a panel of cell pellets and normal tissues, both via in situ hybridization and via immunohistochemistry with the antibody being tested. And ideally, the protein expression pattern will match the mRNA expression pattern. In this example, we're studying protein A expression in a panel of 4 different cell lines that express decreasing concentrations of protein A from left to right. In the first row, we're studying PPIB mRNA expression in these cell lines, and PPIB is a reference gene that we use to assess mRNA integrity. As you can see, all 4 of the cell lines have a robust signal suggesting that the mRNA is in good shape. In the second row, we're studying target A expression via in situ hybridization. And there's a small amount of target A expression on the left panel here, which decreases left to right as we expect. In the third row, we're studying protein expression of target A with test monoclonal antibody. And again, the protein expression is highest in the left lower panel here, and it decreases left to right. So the protein expression pattern roughly equates to the mRNA expression pattern in this example. So the next step in the monoclonal antibody qualification process is to study protein and mRNA expression in a panel of normal tissues. And in this slide, we're studying target A expression by in situ hybridization on the left and immunohistochemistry on the right. And again, ideally, the protein expression pattern will match the mRNA expression pattern. So here in the testis, you can see target A messenger RNA is expressed by spermatids and spermatozoa. And on the right, the monoclonal antibody that we're testing has approximately, or yields approximately the same protein expression pattern. This is the same monoclonal antibody that we had previously evaluated on cell pellets in the last slide. So with the cell pellet data and the normal tissue expression data, we're showing nice correlation between protein and mRNA expression. We qualified this antibody as an immunohistochemistry reagent. We can also use an adaptation of in situ hybridization to track our chimeric antigen receptor T cells in tissue sections. Because the chimeric antigen receptor is a synthetic molecule, we can develop in situ hybridization probe against that molecule that will uniquely identify that sequence in the tissue sections. So this is a map of one of our chimeric antigen receptors with the antigen binding region on the left and the intracellular signaling region on the right. We work with advanced cell diagnostics to generate specific in situ hybridization probes that span these little gene junctions here, thereby uniquely identifying the sequence. In this slide, we're demonstrating the specificity of our CAR-T probe. The top row represents sections of CAR-T cell pellets. And the bottom row represents sections of pellets made with untransduced T cells. The middle column demonstrates in situ hybridization with our PPIB probe, which, as you recall, we use to assess mRNA quality. We can see good quality mRNA in both cell pellets. The right column is our negative control probe, DAP-B, which is against a bacterial protein. You can see we don't see any signal in either cell pellet. And finally, in the first column, we're probing for the CAR itself, and we see robust expression in our CAR T cells, but not our untransduced cells. So this probe looks pretty nice. In this slide, we're using in situ hybridization to identify the location of CAR T cells in an A549 xenograft implanted in an NSG mouse. So the xenograft was implanted, was allowed to grow for a couple of weeks, and the animal was treated with the CAR T cells. We allowed them to traffic for a couple of days, euthanize the animal, fix the tissues, and then perform in situ hybridization. Here we used fast red as the chromogen, and you can see numerous CAR T cells scattered around the tissue section. Now the nice thing about using fast red as the chromogen is that it also fluoresces in the A549 channel. So you can turn this into an immunofluorescence, or a fluorescence image, I should say. And here the CAR T cells appear orange. On the right column here is the same tissues stained with our DAP-B control probe, demonstrating that there's no signal. This is a higher magnification of the same tissue, and here you can see the CAR T cells are roughly aggregating around blood vessels, which is an interesting finding. Now we can also use in situ hybridization to predict end organ toxicity during in vivo studies. And I'll show you some of that data in just a minute. But let's begin with this slide, which compares target X expression in normal human and mouse tissues. The left two panels represent RNA-seq data in human tissues publicly available from the GTEx portal online. And the right two panels represent mouse mRNA expression data publicly available through Bio-GPS. As you can appreciate, there's a significant amount of target X expression in a variety of human and mouse tissues. And according to our algorithm I described a few minutes ago, we would have rejected this target straight away. Compare that with target Y, which has minimal expression in most tissues, and frankly, it's only expressed on B cells. Regardless, to demonstrate the robustness of our target selection algorithm, we ran both of these targets through the algorithm and into in vivo studies. So according to our target selection algorithm, the next step in the target evaluation process is to perform in situ hybridization on a panel of normal tissues, which we did for both target X and target Y. Now, as I mentioned, target Y was only expressed on B cells in select tissues, so I'm just going to show data from target X in the next few slides here. Here's in situ hybridization evaluation of target X in the liver, and we can see expression in hepatocytes, in biliary epithelium, as well as portal vein endothelium. Here's target X expression in the cerebellum. Notice the robust expression in the granular layer neurons, and there's no expression in either the molecular layer or Purkinje cells. This is target X expression via in situ hybridization in the heart. Here we can see expression in cardiac myocytes and the endothelium lining the ventricles. This is target X expression in the kidney, and we see expression in vascular smooth muscle. We can also see expression in the renal tubules, as well as the glomeruli. Here's target X expression in the mouse ovary, showing expression both in granulosa and theca cells, as well as luteal cells. In general, we were able to find target X expression in practically every tissue we looked at in the mouse, but the one that concerned us the most was the lung. Here we see target X expression in the pulmonary vascular endothelium, as well as the walls of the alveoli, which might represent type 1 pneumocytes or possibly microvascular endothelial cells. Now, if you think about it, after treating these animals with CAR T cells, the first large vascular bed they'll encounter is the lung. And with the massive amount of surface area there's the potential for all the CAR T cells to see antigen simultaneously and become activated. So we predicted that the lung would likely be the most important target organ in this particular case. We also performed in situ hybridization evaluation of human lungs for human target X and saw a similar expression pattern. Now, to confirm that target X protein was expressed in the human and the mouse lung, we performed flow cytometric analysis with monoclonal antibody against target X that was a useful flow cytometry reagent. And we studied human primary pulmonary microvascular endothelial cells on the left and an NSG mouse lung dissociate on the right. And as you can see, we can see a small amount of target X protein expression both in the human pulmonary microvascular endothelial cells as well as the dissociated NSG lung cells. We next generated CAR T cells against target X and target Y. As it turns out, the anti-X CAR T cells recognize both the human target and the mouse ortholog. In contrast, the anti-Y CAR T cells only recognize the human target, but not the mouse ortholog. To further demonstrate that target X protein was expressed both in human and mouse lung, we incubated human pulmonary microvascular endothelial cells with the anti-X CAR T cells from two different donors. And as you can see, the CAR T cells recognized protein on these cells, became activated, and produced interferon gamma. Likewise, the anti-X CAR T cells were able to recognize the mouse protein and were similarly activated. Our anti-Y CAR T cells are not activated because target Y is not expressed in the lung. So to summarize, anti-X CAR T cells recognize both the human and mouse target. They can recognize protein expressed both on human and mouse lung cells, and therefore we predict that these cells might cause pulmonary toxicity. In contrast, target Y only recognizes the human target on B cells, doesn't recognize the mouse target, and this target isn't expressed in the lung anyway, so we do not expect pulmonary toxicity with these cells. Now, to test our hypothesis that anti-X but not anti-Y CAR T cells would cause pulmonary toxicity in mice, we performed an in vivo study. We actually turned this into a combination efficacy toxicology study. So we implanted RPMI 8226 cells in these NSG mice about 23 days prior to the initiation of the study. And this particular cell line expresses both target X and target Y. On day 0, groups of mice were treated with either vehicle, anti-X CAR T cells, or anti-Y CAR T cells. And we collected the animals on days 2, 4, and 7 after treatment. We monitored body weights, clinical symptoms, the xenograph burden, and then we performed histopathology on a variety of tissues and studied in vivo CAR T cell distribution and activation by looking at a number of markers, including human granzyme B. We performed in situ hybridization to identify the location of the CAR T cells, and we also studied human PD-1 expression on the CAR T cells. Now, it was interesting. We noted that about 24 hours after treatment, animals treated with the anti-X CAR T cells developed clinical symptoms, including lethargy. Their fur was ruffled. They started losing weight, and a few of the animals actually required euthanasia. But they seemed to start recovering by about 48 hours. Now, we also monitored tumor growth, which is measured on the tumor volume on the y-axis here versus days on the x-axis. And you can see that compared to our vehicle control, our anti-X CAR T cells had no impact on tumor growth at all. In contrast, the anti-Y CAR T cells cleared the tumor. This slide contains photomicrographs of lung specimens from the 3 treatment groups on day 2 after treatment. And here we've stained for human granzyme B to indicate the location of activated T cells. And what you can immediately appreciate is that there's a significant number of activated T cells in the lungs of animals treated with the anti-X CAR T cell. These animals also had acute pulmonary vasculitis. In contrast, the animals treated with the anti-Y CAR T cells had a few random granzyme B positive cells drifting through, but no tissue damage. In this slide, we're studying the location of CAR T cells in the lung by combined in situ hybridization for the CAR T marker and immunofluorescence for CD3. DAPIs, the counterstain. And here you can appreciate robust perivascular and interstitial accumulation of CAR T cells in the animals treated with the anti-X CAR T cells. In contrast, there are very few CAR T cells present in the lungs of animals treated with the anti-Y CAR T cells. Next, we asked if the CAR T cells were activated. So we stained the lungs for human granzyme B via 2-color immunofluorescence. Here, granzyme B cells are red, the T cells are green, and as you can see, the majority of T cells present in this section here are expressing granzyme B, indicating that CAR T cells are in fact activated. In contrast, we see very few activated T cells in the animals treated with the anti-Y CAR T cells. PD-1 is another activation marker on T cells, and in the left panel here you can see again the vast majority of human CAR T cells express PD-1. In contrast, we don't see any PD-1 expression in animals treated with the anti-Y CAR T cells. As I mentioned, we collected tissues on days 2, 4, and 7. And in this series of photomicrographs from animals treated with the anti-X CAR T cells and stained for granzyme B, you can see that the cells were maximally activated on about day 4. So the anti-X CAR T cells were causing pulmonary toxicity. And if you recall, at the same time, they were not causing xenograft regression. So we wanted to see if we could find any anti-X CAR T cells in the xenografts at any time point. And here's photomicrographs of the xenografts from animals treated with anti-X CAR T cells on days 2, 4, and 7, demonstrating there are very few CAR T cells present at any time point. And the insets demonstrate CAR T in situ hybridization. In contrast, if you recall, the anti-Y CAR T cells cleared the xenografts. And if we look at CAR T cell infiltration and activation on days 2, 4, and 7, you can see that by day 7, the bulk of the mass is composed of activated anti-Y CAR T cells, as demonstrated by granzyme B expression. These are additional photomicrographs of the xenografts from animals treated with the anti-X and anti-Y CAR T cells. Here, again, we've used combined in situ hybridization for the CAR T marker and immunofluorescence for CD3 to demonstrate the location of CAR T cells. There are few CAR T cells present in the xenografts of animals treated with the anti-X CAR T cells, whereas, as I mentioned, the entire mass of the xenograft was composed of anti-Y CAR T cells by day 7. To confirm our data, we wanted to repeat the study, and this time we included an additional treatment group here. That is, in addition to animals that were treated with anti-X CAR T cells, we also treated animals with another group of T cells that were transduced with a truncated anti-X CAR. This particular CAR has the extracellular signaling domain, but lacks the intracellular signaling component. So they can recognize antigen, but they don't get activated. These are photomicrographs of mouse lungs collected on days 1, 2, and 4 after treatment and stained for human granzyme B. As we saw in the previous study, animals treated with anti-X CAR T cells have acute pulmonary vasculitis, and we can see granzyme B positive activated CAR T cells present in the perivascular interstitium. Now compare that with group D. These animals were treated with the truncated anti-X CAR T cell, which recognizes antigen but does not signal. And as you can see, there's no evidence of disease in these animals or any of the other control group animals. So in summary, we established an algorithm to help us select CAR targets. To test the robustness of this algorithm, we evaluated target X and target Y. And again, target X would have been rejected according to the algorithm. In situ hybridization, FACS and in vitro CAR T cell activation data allowed us to predict that anti-X but not anti-Y CAR T cells would likely cause pulmonary toxicity. Anti-X CAR T cells did in fact cause pulmonary toxicity, expressed PD-1 in the lung, possibly indicating exhaustion, and did not promote xenograft regression. In contrast, anti-Y CAR T cells did not cause pulmonary lesions and induced robust xenograft expression. And we're currently using this CAR target selection algorithm at Bluebird to select new targets. Next, I'd like to cover preparation and quality control of tissues, cell pellets, and cytospin slides for RNAscope in situ hybridization. This slide demonstrates optimal formalin-fixed paraffin embedded tissue preparation for RNAscope in situ hybridization. So we start with the tissue of interest and obtain a relevant cross-section. And this cross-section should be 8 millimeters and at least 1 diameter to allow adequate formalin penetration. The tissue specimen should then be incubated in 10 volumes of 10% neutral buffered formalin for 24 plus or minus 6 hours at room temperature. And ideally, the tissue after fixation would be immediately processed and embedded. Now, there might be circumstances where you can't immediately process the tissue after the formalin fixation step. And in that case, the tissue can be transferred to 70% ethanol. You can even ship it in 70% ethanol. And then it can be processed subsequently. Now, before you study target expression in a tissue, it's important to demonstrate that the RNA in that tissue section is intact. So here we're looking at 2 different tissue samples, one from a cynomolgus macaque, cynomolgus macaque kidney that was optimally prepared according to the protocol I just described. And then we have a section of human kidney that was collected from a cadaver. Now, if we look at the lower left panel here, we've stained this section of the cynomolgus macaque kidney for PPIB expression via RNAscope in situ hybridization. And if you recall, this is the reference gene we use to demonstrate RNA integrity. And there's a nice signal across the section here demonstrating the RNA is intact. Now, compare that with the human tissue section collected at autopsy. And on the H&E slide, you can see there's a significant amount of autolysis, particularly in the renal tubules. And when we stain the section for PPIB expression, as compared to the macaque tissue, you can appreciate there's little to no RNA expression left. So this tissue would not be suitable for in situ hybridization analysis for a new target. Here's another example. In this case, we're studying cynomolgus macaque ileum on the left and human ileum on the right, collected at autopsy. As you can appreciate from the H&E slide, this section of Cynomolgus macaque ileum has been appropriately preserved. There's nice crisp tissue features here. And when we perform in situ hybridization using the PPIB reference probe, you can appreciate robust mRNA expression throughout the section. In contrast, the human sample is autolyzed and there's no RNA expression or no RNA left. And again, this particular human tissue would not be appropriate for an in situ hybridization study. Now, as I mentioned previously, we use cell pellets as internal controls in both our in situ hybridization and immunohistochemistry studies. And this slide demonstrates the optimal way to make a cell pellet for the purposes of in situ hybridization, and it's also useful for immunohistochemistry. So we start off with about 50 million cells for each pellet. The cells are fixed in 10% neutral buffer formalin at room temperature for 24 hours. After we wash the cells, we add molten histogel and then incubate the histogel in the freezer for about 20 minutes or so until it becomes firm. We then scoop the pellet, the histogel pellet containing the cells, out of the cell into a tissue cassette and fix the cassette in 10% neutral buffer formalin for 2 hours at room temperature. Subsequently, the pellet is processed like a normal tissue sample and embedded in paraffin. In this slide, we've prepared 4 different cell pellets using the method I just demonstrated, and then probe them for PPIB expression. That's our reference gene expression to assess mRNA quality. And here you can appreciate using this technique, there's robust mRNA integrity in all of the pellets. Now, in some cases, you might not have enough cells to actually make a pellet. In that case, you can still study target expression in this small cell population via in situ hybridization by making cytospin slides. So we'll start off in this case with a lesser number of cells, fix them in neutral buffer formalin for about an hour at 37 degrees. Then we'll wash the cells and resuspend them in 70% ethanol, and then spin them onto positively charged slides using a cytocentrifuge. After the slides are dried, we subsequently take them through graded alcohol, graded ethanol, I should say, 50%, 70%, and then 100%. And then we store the slides in 100% ethanol at minus 20 degrees for up to a month. This is an example of a cytospin slide that was prepared as I just described and probed for our reference gene PPIB, and you can appreciate the robust RNA expression in these cells. As you proceed with your studies, it's important to note that each tissue you deal with will have optimal ISH parameters and an optimal probe to assess RNA integrity. It's best to optimize these conditions in your own lab, but a great point to start is the RNAscope reference guide available from ACD at this web link down here. This will give you a good starting point for each species and each tissue. At Bluebird Bio, we use Cynomolgus macaque tissues extensively for in situ hybridization studies, and we've established a series of optimal parameters for each tissue. Now, the variables that are different between the tissues include the antigen retrieval temperature, and it's either at 95 degrees, which is known as standard conditions, or 88 degrees centigrade, otherwise known as mild conditions. And then the 3 probes we use for the different tissues include PPIB, as I've mentioned previously, UBC, and POLR2A. And you can establish a table like this in your own lab for each species. So in summary, in situ hybridization is a powerful technique to rapidly screen through target nominees and identify potential safety risks. Tissues, cell pellets, and slides should be optimally fixed to ensure maximal mRNA preservation. And we should evaluate the tissues by light microscopy and in situ hybridization to ensure mRNA quality before using these tissues in subsequent target expression assays. Primary cells can be used to confirm in situ hybridization results. And because RNA expression may not equate to protein expression, tissue samples should be further evaluated using other techniques to confirm protein expression, such as immunohistochemistry, flow cytometry, Western analysis, and so on. If protein expression is below the limits of detection of these assays, it's possible to incubate primary cells with CAR T cells in vitro to demonstrate protein expression. And in conclusion, I'd like to acknowledge my colleagues at Bluebird Bio who helped me execute all of these studies. Thank you very much.
Thanks, Jim, for that really informative talk. I'm sure our audience now has a much greater appreciation for the versatility of in situ hybridization techniques in streamlining the screening process for potential CAR targets. Before we start the Q&A session, I want to let everyone know that this is their final chance to submit questions for our speakers, so hurry up and send them in now. It looks like we have some really great questions that have come in already, so let's get to the Q&A session. Give us a few moments on our side to get everything situated logistically, and while we wait to transition into the Q&A, the next few slides are some detailed protocols from Jim's presentation and the methods that he used in his research. Don't worry if you didn't catch all the information on the slides. All of it will be available to replay once the presentation is complete.
All right, everyone, so let's start the Q&A. Our first question is going to be for Courtney. Courtney, one of our audience members would like to know, can I quantify the RNAscope results, and if so, how?
Yes, that's a great question. So because of the ability of detecting the RNA molecules as individual dots, they can be quantified. It can be done in a semi-quantitative method according to RNAscope guidelines, which you can find on our website, or you can actually do a quantitative method using digital image analysis platforms, and then you can present your data as average number of dots per cell, or the % positive cells, or the number of cells positive in a region of interest, and so forth. And we have a really nice data analysis guidelines on our website that you can download for more detailed information.
Great, thanks, Courtney. Jim, we have a question for you. One of our audience members asked engineer to have no PD-1, LAG-3, or TIM-3 checkpoint molecules.
Yeah, thanks, Jeff. That's a great question. There are a number of biotech companies and academic groups involved in CAR T research, and everyone's trying to figure out how to maximize CAR T efficacy in the tumor microenvironment. So one strategy is to knock out T-cell regulatory molecules such as PD-1, LAG-3, et cetera, in addition to transducing the cells with a CAR. Now, the challenge is that every time you manipulate the cell, you can have an impact on the CAR T-cell that results. So by knocking out these molecules or doing other genetic manipulations to the cell, you can actually decrease the expression of the CAR. You can decrease CAR activity or decrease the expansion of the CAR population in vitro and not have enough cells to treat your patient. So this is really an area of active research.
Great. Thanks very much, Jim. Courtney, we have a couple questions for you. One came in, says, how long does the RNAscope assay take to perform?
Yes. So we have a couple of different assays. The manual assay takes approximately 8 hours doing the chromogenic, or about 6 hours if you're doing the fluorescent assay, and then a little bit longer for the duplex assay. Now, if you're going to do an automated assay, which we offer again on Ventana or Leica, that can take about 12 hours, but it only requires approximately 30 minutes of hands-on time.
All right, thank you, Courtney. Another question, audience member would like to know, is 2Z enough or is 3Z better for sensitivity?
Yes, so with our probe design, we always aim for 20 Z pairs in the probe for optimal detection, and that will vary slightly depending on your target sequence, but we always aim for 20 double Zs. However, what we show in our technology paper from 2012 is that you really just need a minimum of 3 Z pairs in order to see the signal. So that's with the RNAscope assay. If you're using the BaseScope assay, which I mentioned has a different amplification system, we can get down to as few as 1 Z pairs. So it just depends on which assay the customer would be performing.
Great, thanks, Courtney. And stay with me. We got one more for you. Audience member asked, are there any recommendations for performing dual ISH-IHC assay?
Yes, we do have a couple of recommendations. One is that we really recommend performing the RNAscope ISH assay first, then followed by the IHC assay, and that you also work with a well-validated antibody that you already know works well in your hands. And then lastly, we also recommend that you run the IHC assay with the RNAscope pretreatment reagents first, because the RNAscope assay does require a protease step in order to detect the RNA signal. So you want to make sure that your protein is going to survive that protease step.
All righty. Thank you. Jim, looks like we have another question for you. Our audience member would like to know, they want to know if they can have the cell cytospin and blocks protocol.
Certainly. I provided our sort of generic protocols with part of this presentation, but if our colleague wants to drop me an email, I'd be happy to share those with her.
Great. Thanks very much, Jim. And with that, it looks like we've come to the end of our webinar. I'd like to remind everyone that the webinar will be archived on our site for up to a year at www.gen. So if you missed any parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Jim and Courtney again for their informative presentations, and I'd like to thank the audience for their attention and very thoughtful questions. And a very special thanks to Advanced Cell Diagnostics for supporting this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.